It is a pleasure for me to introduce you my colleague and friend, Dr. Antonio Capalbo, that is Genomics Research Director and Laboratory Director at iGenomics Italy. As I always do, we sincerely apologize for the number of questions that I'm likely not be able to ask to our lecturer. We will do our best to try to answer you in the next weeks. Of course, I also wanted to tell you that as usual, once today's talk is over, in the next few days, you will receive a certificate of attendance and a brief survey of just 3 questions where we want you to share your ideas, doubts, and concerns with us. And without further delay on my part, Dr. Capalbo, please, whenever you want, the floor is yours.
Thank you, Nestor, and welcome everybody to this webinar. Thanks for joining us. And it's a pleasure for me to discuss with you in the next 30 minutes and what are our results from selection clinical trial that we have recently completed. Just a second. Okay, so that we have recently completed about trying to get some evidence-based data about the developmental potential of mosaic embryos, and I will show you our hypothesis, design, and results, and what can be take-home message from a clinical perspective. So first of all, I always like to start my presentation pointing out about the paramount importance of meiotically derived aneuploidies for human fertility. And this is something that we all know well from decades of investigation in human genetics at any stage of development, newborn level, at the prenatal level, and also in pre-implantation embryos. We know that vast majority of aneuploidies, they come following meiotic segregation errors during female meiosis. Now, in this study that was led by Professor Eva Hoffman, you can really appreciate how important are meiotic aneuploidies for human fertility. You see in the red curve, we have the euploidy rate that was derived from direct observation on thousands of oocytes collected across the board of female age. And you can see how this U curve perfectly shape the curve of natural fertility over reproductive lifespan from menarche to the menopause, meaning to say that meiotically derived aneuploidies are really the ones that matter for human fertility and, of course, for a successful IVF treatment. Now, these aneuploidies are inherited from one gamete, and they are present throughout all the embryonic cells and are associated with very known outcomes that we can interpret very well, we can manage, we can counsel patients about. And these are, of course, implantation failure, depending on which chromosome is involved. These aneuploidies can result in implantation failure, miscarriages, or in worst case, in rare cases, they can result in a live birth of a child if it's affected by fetal aneuploidy. Now, often we always hear about self-correction, and I also want to make some clarity at this point here. Self-correction, by definition, is describing that event where a meiotically derived aneuploidy, you have an example here of a trisomy, which is being generated following a disomic, so a normal egg and an haploid sperm. You can see here that you have a trisomy, and the self-correction event during the pre-implantation development, let's assume, means that this triploid zygote will correct its karyotype to a diploid constitution. Now, this event is measurable because in many cases, this event will result in the generation of a population of cells that are characterized by uniparental disomy. And this has been estimated in a very large study on thousands of blastocysts by Nathan Trev back in 2014, showing that UPDs is extremely rare at the blastocyst stage, accounting for 0.06% of all the chromosomes that we find when we analyze the blastocyst stage. This is clearly showing that self-correction is usually very, very rare, and meiotically derived aneuploidies, when they are present into the zygote, they are fixed throughout development and associated with very clear outcomes. Now, a way to understand what is the fate of uniform aneuploid embryos is the best way is to run prospective non-selection studies. In this type of study, you can see, and you may have seen in the study that was recently published by Tiegs and colleagues in 2020 in fertility and sterility, you can see that the typical design of a non-selection trial is that you take the biopsy, you save it for later analysis, and you transfer the embryo without any information about its genetic constitution. So it's in an unknown aneuploidy status. After you have observed the clinical outcomes, then you can analyze the sample, the biopsy that was saved, and you can derive positive and negative predicting values of an aneuploidy diagnosis. In this study, that is the largest published so far about the transfer of uniformly aneuploid embryos, they have transferred 102 embryos that were uniformly aneuploid, so likely of meiotic origin, and they saw that none of them result in a live birth, confirming the fact that when we have and when we diagnose in Pgta cycles uniform aneuploidies, this prediction is extremely accurate, both at the genetic level and also in predicting negative clinical outcomes. Now we have another set of data where aneuploid embryos have been transferred. This was not a non-selection trial, but was a kind of collection of clinical experience from the group of Norbert Glaser, that, as you know, is not the biggest fan of Pgta. And in his experience here, you have in an autocorrection when they did these amendments, they show that out of the 61 fully aneuploid embryos that were transferred, none of them result in a live birth, but some of them also result in a miscarriage with the same karyotype that was predicted into the Pgta cycle. This is also useful as data because these outcomes are coming from different genetic laboratories and are telling us that in general, the prediction of uniform aneuploidies, meiotically derived aneuploidies, is quite consistent and robust across laboratories, and the outcomes are almost certain, even in those clinics that are not big fan of Pgta. And here in this slide, you can see a collection of transfer of uniformly aneuploid embryos that we have recently compiled with my friends and colleagues. And you can see that today we have over 300 embryos that were transferred in the fully aneuploid category. And very, very few of them, 99% of the cases, these result in a failed implantation or, worst case, in miscarriages. So today, I think we have enough evidence to support the fact that deselecting uniformly aneuploid embryos from Pgta is extremely accurate as a way of, is a very accurate diagnosis, and also is no doubt avoiding negative outcomes to patients. Now, it is extremely important that we make distinctions across the broad spectrum of aneuploidies. So mosaic embryos are, of course, another category, but uniform aneuploid embryos, we know today that they are for sure designated in the right way and should not be transferred back to patients unless these data are clearly shown and unless an ethics committee has approved this type of medical practice. Now, let's get to the point of mosaic embryos and the clinical outcomes associated with them. First of all, I'd like just to make some clarity about what is the definition of mosaicism. Usually, this refers to the presence of genetically distinct cells within an organism, in our case, within the embryo, that result from post-zygotic mutational event. Mosaicism can be of two main categories. One is the genetic mosaicism, when we have different population of cells with differences in a DNA sequence, such as different CNVs or indels. Or the mosaicism that we usually discuss in Pgta is the cytogenetic mosaicism, where we have karyotypically distinct cells in one embryo. Now, further to that, we can see that we have also different types of cytogenetic mosaicism. One is the aneuploid mosaicism, where all the cells are abnormal. They can be because there is a mitotic aneuploidy on the top of meiotic one. And in this case, it's not posing that many challenges for diagnosis, because in any case, all cells are abnormal. Or the ones that is concerning us the most is the diploid aneuploid mosaicism. It is a mix of normal euploid cells and abnormal cells. Now, another myth that we often hear in meetings and in congresses is that since we are all mosaics and we are accumulating evidence that in the adult stage we are all mosaics, also all embryos, all pre-implantation embryos should be mosaic. This is absolutely not based on any evidence. We know that, of course, the more we grow, the more we age, the more our cells become different. Of course, in general, the incidence of mosaicism is directly linked to the number of mitoses that we have had in our life cycles. Now, the point here is that at the blastocyst stage, of course, this is the stage where the lowest number of mitoses happen as compared to the prenatal or the postnatal stage. So there is absolutely no reason to believe that the blastocyst stage should be subjected to a higher mosaicism rate, or to translate information that comes from the postnatal, from the adult stage, to this very early stage of development, that is the pre-implantation embryo. Another important information that we should bear in mind is that mosaicism is not increased during IVF procedures. So pre-implantation embryos during Pgta treatment cycle should not be at higher mosaicism rate as compared to spontaneous pregnancies or conception. We have different studies showing that both mosaicism rate, both CBS or in amniocentesis, they are similar in terms of prevalence and incidence. And indeed, infertility and IVF procedure do not seem to increase the incidence of chromosomal mosaicism in IVF-derived pregnancies, meaning to say that we are not under such a big pressure to improve our diagnostic capabilities in Pgta treatment cycles for detecting mosaicism, because there is no increased risk as compared to spontaneous pregnancies, and in general, the incidence of mosaicism in pregnancies is quite low. Now, going more on the technical consideration, how do we define and designate mosaicism in Pgta cycles with next generation sequencing? So the typical approach is that we take a trifactorial biopsy that can be made up of different cells from 2 up to 10, 15 cells. And then we run an NGS analysis, usually a shallow sequencing, and we define and derive based on the reads that we obtain on each chromosome. We derive the intermediate, the copy number values. So we can set some thresholds, then euploid threshold, the aneuploid threshold, and those chromosome copy number values that fall between the aneuploidy and the normal range, they are usually designated as mosaic. Now, there are several assumptions behind this way of classifying mosaicism that are worth to be commented. The first assumption is that with our assays, we can never distinguish between technical variability and biological variability. So if we have noise or experimental challenges or issues in our analysis, we will never be able to distinguish whether an intermediate copy number value is coming because of a technical artifact or through biological variation. And this may happen into the clinic because we are exposed to a high variability of conditions. We know that biopsies can change in sites, that may have different cellularity, may have different quality. We also change reagents over time in our laboratories, and these may have different performances. And all of these need to be accounted when we try to develop algorithms and classification criteria for aneuploidies. Another important consideration to make, I think, is that usually the mosaicism thresholds are defined based on validations of cell lines. We mimic mosaicism by mixing euploid and abnormal cells in different proportions, and then we set thresholds based on the outcomes and the profiles that we get from this type of analysis. which is, of course, very different from what happened in the reality, in the clinical scenario. Now, these cells, of course, are very stable. This model is extremely reproducible and is not really mirroring what happened in the clinical practice, where we are exposed to a higher level of variability. And indeed, if we look at the experience, worldwide experience of reporting mosaicism, we can really see different rates of mosaicism, extremely different rates of mosaicism being reported by different clinics. And of course, I think we can come easily to an agreement that it's not the biology which is different from one clinic to another, but more a kind of way on how these thresholds are established, how technologies are validated, and how the procedures are standardized and monitored over time. The second very weak assumption behind classification of mosaicism is that by definition we will be always subject to the sampling bias, which means that if we define a percentage of mosaicism, one biopsy, this doesn't mean that this will be the same to the embryo, the resulting embryo. Meaning to say this type of analysis will always be subject to sampling bias, and by definition, we won't be able to predict with enough accuracy what would be the mosaicism rate into the embryo, in the whole embryo, by the analysis of a single sample. However, we know that when we report mosaicism, or even when it's possibility when we overcall mosaicism in the Pgta clinical setting, this comes with some very important consequences on patients. So first of all, they have to face the decision on whether to transfer or not to transfer these embryos that they are in any case classified as abnormal. And this will generate a course of actions that are really important for knowledge, such as they should go through an additional session of genetic counseling in order to evaluate whether or not to transfer these embryos, and also they will experience for sure a higher anxiety, distress, and higher costs associated with this procedure. Then transferring mosaic embryos after Pgta has generated this kind of indication in some cases for amniocentesis, for undergoing invasive prenatal follow-up, which should be justified because this comes with very well-known risks of iatrogenic abortion, as we all know well. But I think the worst outcome on reporting or overcalling mosaicism in our field is the potential wastage of otherwise healthy and normal embryos. For instance, by looking at one of the largest experience on the transfer of mosaic embryos and reporting of mosaic embryos, Monet and colleagues in 2017, they showed that less than 3% of the almost 5,000 of embryos that were classified as mosaic, then they were transferred back to patients. That means that unless we have very robust data suggesting that these embryos bring some risk or they have a lower reproductive potential, this may really cause a significant wastage of normal and otherwise viable and healthy embryos. So what is the evidence that we have to support this practice of discarding or downgrading mosaic embryos? Now, most of the data and the experience was coming from retrospective studies, from retrospective analysis, where mosaic embryos are transferred as last option usually. And what they found in this largest paper by Biotti and colleagues is that low mosaic embryos, up to 50% of intermediate copy number variation, they had a slightly lower live birth rate as compared to the uniformly euploid embryos, while the high mosaics, they behave significantly different. They show a significant reduction of reproductive potential, although no mosaic pregnancy or no aneuploid pregnancies has been reported in this retrospective analysis. But what is the most important bias when looking at mosaic embryo transfer in retrospective study? The most important bias is the patient selection. So what happened in the typical workflow of a Pgta cycle when mosaicism is reported is that euploid embryos are transferred as first option, while mosaic embryos are left behind. And as we all know well in IVF, good prognosis patients usually get pregnant with first or second embryo transfer, while poor prognosis patients are the ones remaining at the end, and they are the ones where we are measuring the reproductive potential of mosaic embryos. So in this case, by looking at mosaic embryo transfer data in a retrospective fashion, we are really introducing a significant population bias. If you look at the Biopti studies, indeed, you can see that 95% of cases, a mosaic embryos was selected for transfer to a patient when no euploid embryos was available. So in order to avoid this population selection bias, we run a prospective non-selection trial. The main difference with retrospective studies is that in this case, we have not reported the diagnosis of mosaicism back to the clinic and to the patients. So the outcome of mosaicism was blinded, double-blinded to physician and patients. And in this way, we had the selection of we did not bias the selection of the embryo that was based on standard criteria such as morphology. And the selection of embryo transfer was thus random for the genetic outcome. After the embryo transfer occurred, then we have blinded the results and we have generated two main categories of two main study arms, such as the euploid, the uniform euploid embryos, and the mosaic embryos that we have further stratified in low mosaic between 20 to 30% and moderate mosaic between 30 to 50% of intermediate copy number variation. After that, we could also perform some prenatal and postnatal cytogenetic follow-up that I will discuss later. So just to recap, euploid embryos and low mosaic, medium mosaic grade embryos were all reported back as euploid, and the selection was not biased by this genetic outcome. So we could get, I think, a very reliable comparison between these three categories. So I think an important preliminary finding of our study was how to classify embryos into the low mosaic or medium mosaic category, and why we felt comfortable in using these embryos clinically and not reporting back this type of abnormality. So the first step of our study was to disaggregate 73 blastocysts donated for research in 5 different portions, 4 trophectoren biopsies and the ICM. And in this way, we could assess what is the positive predicting value across the broad spectrum of aneuploidies that we can detect in a single trophectoren biopsy. So here you have an example of a low mosaic copy number value, which is consistent with a low mosaic that was not confirmed in the subsequent biopsies throughout the embryo, while here you have a profile plot of a trisomy 21 embryo biopsy that was indeed uniform and confirmed throughout all the biopsies. To summarizing all this data coming from hundreds of blinded biopsies, I will try to walk you through to this table. From in the left side, you have the reference trophectoren outcome, and going to the right, you can see if this abnormality or this normal chromosomal profile was confirmed in the additional trophectoren biopsies, and whether or not the ICM also was involved. For instance, for the uniform aneuploid classification, you can see that 98% of cases, the abnormality was present throughout all the embryonic sessions, confirming the likely meiotic origin. In the high mosaic range, we indeed identify a kind of gray zone where some true mosaic case may be present. You can see here that when we find a high mosaic, 65% of the cases, the abnormality is spread throughout all the embryo, while in other 35% of the cases, this is confined to few portions of the trophectoren, representing and highlighting probably a true mosaic case. So these outcomes can also explain the reason why in retrospective analysis, such as in the Biopti study, the high mosaic embryos show a significantly lower reproductive potential, because they also is a possibility that can be fully unemployed to start with. Then, why we felt comfortable with the transfer of low and medium mosaic embryos, because we observed no statistical difference in the conformation rate or in the abnormality rate when we followed up the whole embryo. In other words, what we saw was that euploid, low mosaic, and medium mosaic embryos had the same euploidy rate throughout the embryos and also in the ICM. So here you have the table of the patient that we recruited in this study, 783 patients performing 824 IVF cycles with Pgta. We had an average euploidy rate of 58% in this group of women with a mean female age of 37.5. And these are the main outcomes that we observed between these three categories of euploid, low putative mosaic, and moderate putative mosaic embryos. As you can see, they are really equivalent in terms of positive predicting positive pregnancy test, biochemical pregnancy loss, miscarriage rate, and live birth rate. We reach our planned sample size, and we also perform multivariate logistic regression analysis, confirming the lack of any association between the Pgta category and the clinical outcomes, miscarriage and live birth rate. And we also failed to detect any difference in neonatal and obstetrical outcomes from pregnancies resulting from euploid or mosaic embryos. We've been also trying to develop some additional metrics in order to further explore any potential association between mosaicism and clinical outcomes, like mean mosaicism rate, mean number of mosaic chromosomes, complex mosaic, more than 3, more than 5 chromosomes, but we failed to detect any association. Here we have just a visual representation on the apparently or allegedly abnormal embryos that we transferred. You can see that putative mosaic embryos for all chromosomes have been replaced over the course of these trials, including trisomies for chromosome 21, 13, and 18. Next, we could follow up pregnancies, particularly in the prenatal stage. We could collect some data of amniocentesis and CBS that were performed spontaneously by patients, because we did not indicate amniocentesis or invasive prenatal diagnosis during the course of this study. So the patients, they decide themselves whether or not to undergo a follow-up, a prenatal follow-up. And the only case of confined placental mosaicism that we observe in the prenatal, at the prenatal level, was coming from the euploid embryo transfer, while we did not detect any of these events for the putative low and medium mosaic embryos. Next, we could perform some postnatal follow-up, cytogenetic follow-up. In this case, with Professor Eva Hoffman, we work on trios. We collect the saliva from newborns and the parental DNA, and we run high-density SNP arrays in order to check for presence of mosaicism or any byproducts such as the uniparental disomies. We did that for a limited number of families because it's extremely challenging, to be honest, to collect this type of samples and to get families involved after the delivery. But in this 38 family, we failed to detect any instance of mosaicism or UPD in newborns from putative mosaic embryos. And finally, in our trial, we also did some modeling in order to investigate what can be the impact of discarding these embryos in terms of cumulative live birth rate, meaning to say how many, to what extent we can impact the pregnancy chance of patients from an intention to treat perspective. So in the situation where we exclude both low and moderate mosaic embryos, we observed that over 1/3 of the babies would not have been born. And this is really highlighting how detrimental this can be in reducing cumulative live birth rate if we discard these embryos or for any case these embryos are not replaced in Pgta cycles. So our findings are also, of course, corroborated by independent evidence. For instance, here you probably are aware about this paper by Mina Popovic and colleagues, where they have followed up the results of a clinical trophectoderm biopsy on embryonic outgrown day 12 of development. And as you can see here, when they got uniform euploid or aneuploid findings, these were confirmed in all cases, while the positive predictive value of mosaic diagnosis, also few days of culture after the biopsy, was extremely low. How to explain our result? Of course, we may acknowledge that these intermediate copy numbers that we see are real, and the few abnormal cells are confined to a small portion of the trophectoderm and get lost during early stages of post-implantation development, similarly to what has been reported for mouse model by Ellen Bolton and colleagues. And also, as it has been supported by independent evidence about the existence of early developmental bottlenecks in human placental that can be able to normalize post-zygotic mosaic aneuploidies in human embryos. As an alternative explanation, of course, we also have to acknowledge that some of these intermediate copy numbers may just be the results and come as a consequence of some technical variation, which is normal when working under clinical conditions. What is important also for us to know and to discuss is is there any risk in pregnancy when we transfer mosaic embryos or putative mosaic embryos? In this table, you can see a collection of data that was recently published by Nathan Treff and colleagues. And you can see that they are actually putting all together the embryo transfer outcomes and how many times and how many cases the same mosaic finding observed in embryos was confirmed in later stages of pregnancies or in the newborn. And this was quite easy because it was only one case in the medical literature where we had a confirmation of the same mosaic finding in embryos and also in the newborn. Of note, of course, this newborn was also free of any phenotype and it was healthy. So in total, we have today the transfer of over 3,000 putative mosaic embryos, and only one of them has been confirmed in a mosaic pregnancy. So this rate is similar, if not even lower, than what is the expected iatrogenic abortion rate that we know is associated with invasive prenatal diagnosis. So I think all this data should be used to counsel patients about whether or not to follow up the transfer of putative mosaic embryos in prenatal with invasive prenatal diagnosis. Negative predictive value is also another important parameter to review. This defines how many times, to what extent we can lower the incidence of mosaicism in pregnancies by avoiding the transfer of mosaic embryos or by transferring only uniformly euploid embryos. This will take a huge sample size in order to be properly investigated. So at present, this is an outcome that is unknown. We don't know if by transferring euploid embryos only, we are able to lower that 0.3% of mosaicism incidence in pregnancies, but we have some evidence about the opposite. For instance, in this paper that you can see at the bottom of my slide, you can see that half of the product of conception that were analyzed following the transfer of uniform euploid embryos, even after high-resolution NGS, they showed mosaicism. So it means that even if we replace only euploid embryos, we cannot make sure, we cannot ensure to our patients that they won't have a mosaic pregnancy. And this, of course, makes all the sense. So my final slide is about how to manage embryo selection also in the context of morphology and mosaic findings. We know that there were some scientific societies suggesting that first choice should always be for transfer euploid embryos over mosaics without any consideration about morphology. I think there is definitely no evidence today to recommend this. morphology should be part of the evaluation, because there is no study today that is showing that transferring a bad morphology, bad quality euploid embryos is a better solution for patients than replacing a low putative mosaic embryos of better quality. So this is, I think, very important to be considered. So take a message. I hope it was quite clear that intermediate copy number in clinical trophectomy biopsies are either technical variations, or they may show and represent true mosaicism that is confined to small trophectomy portion without ICM involvement, most of the cases, and implication for the pregnancy. Reporting this type of mosaicism, or even worst case, overcalling mosaicism, has no elements of clinical utility, while it can result in a significant wastage of viable and otherwise healthy embryos. And this is something that I hope that our data from a prospective non-selection trial will help to avoid, or at least to improve decision-making of patients with more evidence. And I think that also it's important to underline that Pgta assay should be validated in prospective non-selection trials. We should really work toward this direction when we are willing to implement new criteria and new algorithm for classifying aneuploidies or whatever other genetic alteration. We should really work to get first positive predicting values and negative predicting values before implementing them in routine clinical practice. I want, of course, to acknowledge all the colleagues from iGenomics for organizing this webinar, and all the colleagues that have participated in a non-selection trial and that have shared this, I think, wonderful experience. Of course, I'll be happy to discuss further with you to answer any question you may have.
So thank you very much, Antonio. A wonderful presentation, really clear as always. And we do have already some questions. I'm asking also the audience. We have around 200 people live right now, so please keep sending your questions. We will try to do as much as possible. First, you told us at the very beginning of the lecture, but could be that some people miss it because there are a lot of people asking about what do you think about self-correction in embryos?
Well, I think it's good to remark this again. Self-correction, again, is when we have a zygote, usually the best definition of self-correction is when we have a zygote that has a meiotic aneuploidy. So this is expected to be present in all the cells of the ensuing embryo. And the self-correction happens to restore a normal disomic or diploid configuration. This is something that we can measure. We can measure the frequency on this event, on how this event happened in human preimplantation embryos by looking at byproduct of this event, which is the presence of uniparental disomy in a population of cells of the developing embryo. And this has been done on thousands of blastocysts using SNP arrays, and the frequency of UPDs, that is a byproduct of self-correction, is the result of the self-correction, has been found to be extremely low, extremely rare, 0.06%, if I'm correct. Meaning to say that self-correction is not so frequent. When a meiotic aneuploidy is present in an embryo, this is expected to be fixed and through development, and this is associated with very well-known negative clinical outcomes for patients.
Thank you, Antonio, because there were another question related with this, asking, I didn't understand what the 0.06% of blastocyst node was in reference to. Could you explain it again? So you already replied that question. What are the risks of mosaic trisomy 21? And how should we counseling being made for those?
Well, I understand. Yeah, I got the point, and this is indeed an ongoing conversation. I mean, of course, if we break down our clinical outcomes per chromosomes, we do not get enough sample size in order to make robust conclusion on a per chromosome base. I think that the principle that apply to all chromosomes and to all aneuploidies is that unless we don't have evidence that genetic finding, that genetic signal translating something that is clinically relevant, we should consider it at the same strength as for the other chromosomes. So technical variation or true mosaicism may be present, but then may not be clinically relevant or clinically useful as information to report. Of course, we need more follow-up data. Patients will need to be counseled in a very fair and unbiased way. They should be aware about what are the data, what are the evidence around this classification, and they should also be part of the decision-making process. However, as a laboratory perspective, I think that the same concept for me applies to all chromosomes. So as long as I don't have evidence that the findings is abnormal and is translating in something that is not providing a clinically useful information, I'm considering it at the same strength for other chromosomes.
Thank you very much, Antonio. There's a lot of questions that are looking for, your opinion and trying to understand what do you think about this mosaicism, about that, that is a hot topic in our field right now. And they are asking also that you already mentioned at the lecture, but do you think that some type of mosaicism could be a technical artifact?
Yeah, I mean, in the general, let me zoom in and zoom out a bit. So in the general context of Pgta, what really does matter is to classify correctly, to distinguish very well, and deselect the uniform aneuploid embryos. These are the vast majority of the aneuploidy, and these are the ones that we can manage, information we can manage, we can counsel patients with very clear data and outcomes. And we are sure that by deselecting these embryos, we are doing something really good to improve IVF outcomes of infertile patients, in particular for advanced maternal age women. Now, technical variation is something that happens, is something that we should acknowledge, that we should consider, that we should be honest and be fair and account in our model that we develop, in our threshold that we develop for calling aneuploidies. So we should definitely account this type of variation and try to develop a diagnostic approach, which is also contemplating the possibility that there is some sort of level of technical variation. And this can be done. I mean, it's not something that is going to challenge the whole concept of Pgta, or is going to challenge the delivery of our genetic results. As I said before, most important thing and the really thing that matters is deselecting uniform aneuploid embryos from transfer.
Thank you, Antonio. So the questions keep coming, and I guess that there is somebody that goes to PDIS in a couple of weeks in Berlin, because it's asking about, are you going to PDIS? Do you think that it will be before and after this Congress in relation with mosaic embryos and how we think about those embryos?
Yes, I'm going to the PGDIS, and I'll be happy to meet you there. The question is if there will be a discussion around mosaicism at the PGDIS. Of course, yes. This is an ongoing discussion from years, and of course we have seen different perspectives that may be guided by different considerations, and I think today also we start having more agreement in the way that everybody is recognizing today is coming to an agreement where these, at least the low mosaic embryos, which account for the vast majority, the high mosaic is just a small part of the whole embryos that we get, the whole diagnosis that we get at the blastule stage. So I think there is enough agreement today that these embryos shouldn't be discarded. We should not advise new IVF cycle recycling to patients that have already mosaic embryos available. I think the results of our study together with other studies are clearly showing that these embryos are capable of normal development, they have similar reproductive potential, and in particular, most importantly, they do not bring additional genetic risk for pregnancies, as now we have kind of large experience about that. Of course, this shouldn't stop us to keep working on this topic, to keep collecting data, possibly in a controlled way, in prospective trials, and deliver even more stronger recommendations and guidance on what to do with these embryos.
Yeah, thank you, Antonio. And according with this question, there's another ones or two or three, but to arrive to the same point that said we were afraid to transfer mosaic embryos. So should we be afraid right now after the data that you showed us? I guess it's about proper genetic counseling, right?
I think we should be afraid to discard these embryos, not to transfer them. So I turn it the other way. We should be afraid to discard embryos for any reason, for reasons that are not supported by evidence today. We don't have any evidence today showing that these embryos should be discarded or not considered at the same strength as euploid ones. So of course, Nasser, I need to say that this consideration may also change from assay to assay, from technology to technology, and it's really important that people develop in our field and perform prospective non-selection trials in order to establish clinically how the prediction on trophectomy biopsies works. And it is extremely important then for counseling patients and of course for improving the decision making for these embryos. So really like that all technologies, all platforms will have data from clinical trials and prospective non-selection studies in order to validate these assays and these technologies in clinical setting. It is the best way to deliver evidence-based data to patients and physicians.
Glad you say that, Antonio, because the next one was, what do you think about the need of doing prospective non-selection trials to validate the Pgta assays, even more for any new algorithm being implemented? So I guess that you already replayed that it's important, right?
Yeah, it is. And we shouldn't, I hope we won't make this mistake again, where for years now this embryo has been discarded without having proven that they were bad, that they were resulting in higher risk for pregnancies. So I think that, and I hope that in the future, new technologies will be validated first and then applied clinically in case they provide elements of utility and clinical utility. Yeah.
Okay, perfect. Thank you, Antonio. And by the way, I'm removing the part of congratulations and thank you very much for the great lecture. So, but all of the questions start with this, okay? Just to be sure that you know. Somebody's asking, is it correct that invasive procedures are excessive for pregnancies after mosaic embryo transfer? Should we stop to offer invasive diagnostics for these patients? Thank you.
No, that's a great question. Again, what's the evidence for giving an indication of invasive prenatal diagnosis that brings risk for the pregnancy when there is no evidence about increased risk for mosaicism following the transfer of these low mosaic embryos, low or medium mosaic embryos? I mean, I try always to put myself on the perspective of patients, and of course I would like to be counseled, but I would like also to be not to be scared about that, to be provided with clear evidence about facts. So we know that today same consideration, same gestational risk are applied for mosaic or euploid embryos. So why should be an indication for invasive prenatal diagnosis? So is there an increased risk for doing that? No. So why this should be an indication for doing something that is invasive? Then, of course, it's the patients. There are many factors in this decision making for amniocentesis, but the fact that we transfer a low medium mosaic or a euploid embryo shouldn't be a factor here. So we should follow same consideration as for the transfer of euploid embryos.
Yeah, I guess with more and more data, we are being more confident that we should avoid maybe those invasive procedures that at the end can end into a miscarriage after to get pregnant the patient, right? Okay.
Yeah.
So
yeah, at the end it is also, I mean, a patient decision. There may be many different reasons to do amniocentesis, and we should support a fair decision making.
Sure. There's another direct question. So finally, what do you suggest to do? Don't inform about the Mosaicism lower than 50%?
Yeah, I mean, I would call these embryos as euploid, as we've been doing in the trial and as we are keep doing here. And so I really don't see any clinical utility in reporting this, and I would avoid the situation where these embryos are then discarded from clinical use, because they can really add to the cumulative laboratory. They are showing similar reproductive potential and not increased risk. So I really don't see any advantage or any clinical utility in today reporting mosaicism as it is done. So below 50%, for me, they all go to the same umbrella. And of course, it's important also to acknowledge that especially when you're treating a patient population of advanced maternal age, you saw in our trial 37, 38, the mean female age is extremely important. Of course, to deselect uniformly aneuploid embryos that are significant number of the ones that are produced, but it's equally important not to impact the cumulative laboratory, not to discard embryos that are potentially viable and that are potentially the last option for this, especially this type of patients.
Thank you again very much, Antonio. We are almost at the end of the webinar, but we keep receiving some questions. So I'm gonna go straight for some of them. There's a comment in here saying in the future it's very important to follow up this baby's child in long term to verify if there is no real impact from the Mosaicism in the health in a long term. I guess that this is something similar to any baby after an IVF treatment, right?
Yeah, absolutely. Yeah, I was about to say the same. All follow-ups, long-term follow-ups are welcome for every procedure that we perform in IVF. Of course, it would be nice also to have more systematic studies comparing prenatal and postnatal outcomes following the transfer of euploid and putative or apparently mosaic embryos, maybe expanding the cytogenetic investigations, and also, of course, keep monitoring all the outcomes. The point is trying to do this by proper study design, where outcomes and results are controlled, and where all the outcomes are monitored and reported, as it is usually done in clinical trials. Retrospective analysis often lack information, are always biased, as I showed before. So we really need to have to put more efforts in developing this systematic prospective trials.
Thank you very much, Antonio. somebody's asking here, to your knowledge, is there any relationship between Mitoscore and Mosaicism, mitochondrial DNA and Mosaicism?
Now that I'm aware. No.
Okay,
No.
and is there any evidence? Okay, Is there any evidence regarding Mosaicism and maternal age during egg retrieval?
Again, these are good questions. As we see in many studies, it seems that these intermediate copy number values, which is slightly different than saying mosaicism, are not dependent on female age, and these can be explained equally with the fact that this is technical variation, so it doesn't change with advancing female age, of course, or whether it is a different biological phenomenon as compared to meiotic aneuploidies, which is not impacted by the aging of the oocyte. But as long as I'm aware, in many different studies, this has been consistently reported that this intermediate copy number are consistent with mosaicism are not changing, are not affected by female age.
Perfect, Antonio. So we are about to finish again. So there's any relation between the use of the laser and Mosaicism?
Well, this is also interesting as a question, good question. And yeah, this may fit better with the hypothesis of technical variation. There are some recent studies showing kind of effect of the operator of biopsy on the mosaicism rate, which of course we can believe that an operator is impacting mitosis somehow. So it's probably there can be, and it makes sense to me that there is an impact of the biopsy operator because this is probably some operators are doing performing bad biopsies or lower quality biopsies. This may increase a little bit the noise, experimental noise, and sometimes can be confused and misclassified as mosaicism. So this makes perfectly sense to me. Indeed, I think that really the most important task that we have is to try to set and standardize all the conditions that we have in both IVF laboratory and in the genetic laboratory in order to deliver consistent and robust results with a main focus on detection of meiotically derived aneuploidies and deselecting these embryos. Extremely important, the collaboration and the partnership between laboratories and the genetic lab, trying to keep and all the results constant and robust over time, even when integrating new embryologists, and this is extremely important.
Thank you, Antonio. There's a question that keep coming, so I need to make you. I was wondering how the Mosaicism will be in non-invasive embryo liquid biopsy results. I mean, would be non-invasive embryo liquid results, I guess. Would like to hear your comments.
Well, we don't know. I'm not aware of any robust studies that has addressed this outcome yet. So we don't know. We still have many. We still lack many information about the origin mechanism, how this extraembryonic DNA is released by the embryos, and therefore it is really difficult to answer to this question because we really lack some knowledge about the biological mechanism behind that. But of course, it would be very interesting to investigate in the future what would be the reliability of non-invasive Pgta to detect mosaicism.
Thank you, Antonio, very much. If I might say something, I know that our colleague Dr. Cameron Rubio is working on that, and what they found is that levels lower of 50% of mosaic embryos in the media usually correspond into a euploid trophectoderm. Mosaicism over 50% in the media usually corresponds to aneuploid embryos. And this is something that in the media we are seeing that it's so similar what you are mentioning about mosaicism in trophectoderm biopsies. So I guess that we will go in the right way. Well, we do have still a lot of questions. So if you don't mind, Antonio, I will send through your way in order to try to answer in the next weeks to all the colleagues, or maybe they can find you in a couple of weeks in Berlin for the PGDIS. But I would like to ask you for your golden minute. You know, that the last final remark of this amazing work that you are doing and you show to all of us.
Okay. Thanks everybody for staying for the whole meeting, for the whole webinar, and I hope I've been able to convey some important messages and reassure most of you about the transfer, considering the transfer of these embryos that are designated as mosaic. Yeah, I just really hope, as all my colleagues doing this trial, we just really hope that this evidence and this prospective trial will help patients to improve make a more informed decision about the transfer of these embryos, and to improve the field also for the next developments, trying to stimulate the studies and the development and of trials before introducing new algorithms, new criteria, and that's it. This is my take-home message, and I hope you enjoyed the webinar. And of course, I'm available for any follow-up or any other question you may have after that.
Thank you very much again, Antonio. We received a comment also that an IVF patient with one mosaic embryo, this study and webinar has been so helpful. So thank you so much. And I guess that this is for a patient, and we are glad that it clarifies maybe what she can do with the mosaic embryo. But also, I'm pretty sure, Antonio, that after this webinar, there's some clinics that will change the procedure with the mosaic embryos, so we won't left anything behind. Thank you again, Antonio. Wonderful lecture, and thank you all for being here today with us. More than 200 people live asking questions, and again, I apologize not being able to ask everything to Antonio, but we run out of time, and hope to see you here again, Antonio, probably when your next paper get out, right?
Okay, thank you.
Thank you very much to all of you, and have a great day. Bye-bye.